Hi, my name is Mark Zadie. I'm a PhD student in the Department of Medical Biophysics at U of T, and I'm here to talk about my work on the development of computational tools for evaluating differential protein expression relative to spatial oxygen gradients using imaging mass cytometry. So foremost, I'd like to disclose that I am a consultant for Histoways Incorporated and that I'm receiving grant and research support from TFRI and NSERC. Oxygen concentrations in tissue are influenced through a balance between supply and demand. Supply is governed by perfusion or diffusion of oxygen, whereas demand is dependent on biological processes that govern oxygen metabolism, such as oxidative phosphorylation. Here, we illustrate how diffusion-limited hypoxia can actually confer a radioprotective effect to severely hypoxic cells, often to the detriment of the patient receiving the radiotherapy. Lower levels of hypoxia can also lead to biological changes such as increased aggressiveness, therapeutic resistance, and an overall poor patient prognosis. Having the ability to evaluate histological marker expression relative to hypoxia will pave the way for novel biomarker and therapeutic development. IMC is one such technology that can actually achieve this. Tissue sections are stained with heavy metal conjugated antibodies, which are then ablated and imaged using a cytoff mass cytometer. This allows for highly multiplexed imaging without the limitations conferred by spectral overlap. In our case, we want to essentially identify hypoxia-driven expression using IMC. So we had patients that were given the hypoxia pro Pimonidazole orally, which is detectable through IMC. The glioblastoma tumors were then excised from the patients, and IMC was performed using a panel of markers we suspect to be regulated by hypoxia. We can then examine differential expression of the markers relative to hypoxia. So first, we need to delineate regions of relative hypoxia from normoxia by setting a threshold in conjunction with a pathologist. Next, we perform nuclear segmentation using the iridium-193 intercalating agent, which is very similar to the DAPI counterstain for fluorescence microscopy. The start of segmentation shown here is a pre-trained machine learning algorithm used to segment nuclei, which I've adapted for use with IMC images. We can then set thresholds in conjunction with a pathologist, verifying that our classifications are consistent with what they would manually annotate themselves. And finally, we construct a composite classifier, assigning one or more classes to every segmented cell in our dataset. One way to perform differential expression analysis is by measuring the ratio of % positive cells in the Pimo-positive, so hypoxic versus negative regions. More positive values indicate an overexpression under hypoxia, whereas negative values indicate an underexpression. So from this, we can actually see several promising candidates, many of which can serve as a hallmark of hypoxia in glioblastomas. But as many of these oxygen-dependent markers exist as a gradient rather than a binary expression, setting absolute thresholds may not be a sufficient method for evaluating their expression. Rather, we can compare the distribution of IHC marker intensities across our two areas and use statistical means to delineate whether or not there is a measurable difference across our two populations. Hypoxia has been known to influence the co-expression of numerous genes. So here we actually create scatter plots comparing the intensity distributions of each IHC marker combination within a region of hypoxia shown in orange and relative to normoxia shown in blue. The Spearman correlation coefficients are used to evaluate the magnitude and statistical significance of such associations. Some examples of meaningful co-expressions include that of CD68-NIBA1, which shows a strong correlation independent of hypoxia, GLUT1 and Pimonidazole showing a strong co-localization only in hypoxic areas, and ICAM-SOX2 showing a moderate co-localization in the absence of hypoxia. We can further simplify the marker co-localization pair plots by visualizing the Spearman correlation coefficients as a heat map. Sorting the genes by their correlation with Pimo, we can see that many known HIF targets are clustered in the top left corner, whereas those that are negatively correlated are seen in the bottom left. So in short, we present an end-to-end workflow for analyzing hypoxia-dependent expression in IMC images. Some of the ongoing developments include vessel distance analysis, evaluation of hypoxia-associated texture features, and IMC visualization tools for pathologists as shown on the right. Thank you for your time.